In this video, we're going to talk about pyruvate decarboxylation. And what I want you to be able to answer when you're done watching this video is, what are the components of the pyruvate dehydrogenase complex? Where in the cell is it located? What are the products and the reactants of pyruvate decarboxylation? For each of the 5 cofactors in the PDH complex, I want you to know the following. I want you to know its full name. I want to know which enzyme it is a part of, recognize the functional part, and recognize the structure of the cofactor, recognize what it carries, and finally, is it a stoichiometric cofactor or a co-substrate, or is it a catalytic cofactor? Is it bound to the enzyme? I want you to list the reasons for enzyme complexes, and I want you to be able to describe how the PDH complex is regulated. All right, so the PDH complex, the pyruvate dehydrogenase complex, contains 3 enzymes, and they have fancy names, but you only need to know the easy names: E1, E2, and E3. And multiple copies of each one of these enzymes exists in a large complex called the PDH complex, or the pyruvate dehydrogenase complex. And the ultimate reaction that is catalyzed by this complex, by these 3 enzymes, is to oxidize pyruvate into acetyl-CoA and CO2. Basically, what's happening here is we're activating the acetyl group of pyruvate by adding this coenzyme A molecule to it that very much increases the energy of pyruvate and makes it able to be involved in the next set of reactions in the citric acid cycle. And I just want to remind you that the origin of pyruvate, if we're looking at the origin of pyruvate from glucose, then here are the various carbons from glucose. This methyl group is carbons 1,6, 2,5 here with the carbonyl, and 3,4 with the carboxyl group. And so through this reaction, these carbon 3,4 from glucose are going to be gone, are going to be released as CO2, leaving us only the carbon 1,6 and the carbons 2,5 from glucose here in acetyl-CoA. Now, the pyruvate dehydrogenase complex, these 3 enzymes, have a total of 5 cofactors. Now, if you'll remember, cofactors are non-amino acid organic molecules that are necessary for the function of specific enzymes. There are catalytic cofactors, and those are the ones that are actually going to be bound to the enzyme. Like thiamine pyrophosphate, or TPP, is in E1, lipoate is in E2, and flavin adenine dinucleotide, or FAD, is in E3. In addition, there are 2 stoichiometric cofactors, or sometimes called co-substrate cofactors. These are cofactors that are not bound to the enzyme, that they are free floating. And so we have coenzyme A and nicotinamide adenine dinucleotide, which is part of E3. Coenzyme A is involved with the enzyme E2. Now notice what's interesting is that each one of these cofactors is a vitamin or a mineral that you would get through your diet. And in fact, many breakfast cereals are actually fortified with these. So here's your thiamine, that's TPP. Here's your riboflavin, that's the component necessary to make FADH. Niacin, necessary to make NADH. Pantothenate, that's necessary to make coenzyme A. Right? So if your diet, these molecules, these cofactors are so important for the PDH complex, and the PDH complex is so important for general metabolism. If you have a deficiency of any of these, it's going to cause some pretty global symptoms. Okay, so let's start at the start. Here is our 3 enzymes. This is E1 in yellow, this is E2 in green, and this is E3 in red. And then we're going to go through steps 1, 2, 3, 4, and 5. The first step is here in E1, where thiamine pyrophosphate of E1 decarboxylates pyruvate. So here is E1, here's pyruvate, and we attach the acetyl group. So this carbon here of the methyl and this carbonyl carbon gets attached to PPP, and we release CO2 as a byproduct. Boom. And remember, this is multiplied 2 if we're talking about the stoichiometry per glucose. Now that's done by thiamine pyrophosphate. Here's what thiamine pyrophosphate actually looks like, and this is the part of the TPP molecule that actually is going to be carrying that acetyl group. And there it is right there. So here we have our 2 carbons, what's left of the pyruvate. Remember, CO2 is gone. Next, lipoate of E2 now accepts that acetyl group from TPP. It's sort of like the swinging arm mechanism where the lipoate molecule is this long carbon chain that is linked to a lysine, which is also a long carbon chain. So we've got this long swinging arm that starts off in a oxidized state, becomes reduced, and then grabs that acetyl group from the TPP of E1. So let's take a look at what lipoate looks like. Again, not only is lipoate itself a long carbon chain, but it's connected to a lysine, which is also a long carbon chain. And so that is one of the examples of structure gives rise to function. It has this long arm structure, and that's necessary because its function is to swing as an arm, swing from one active site to another active site. Here it is, and this is the functional part of lipoate. Here it is now in its reduced form, and then here it is now holding on to that acetyl group, the 2 carbons from pyruvate. And right there, this sulfur carbon bond, that's called a thioester bond, high energy bond. Very reactive bond, and you're going to see examples of thioester bonds quite often within biochemistry. Okay, next, that acetyl group that is being held by lipoate in E2 is going to be transferred to a coenzyme A. Okay, so now there's our finished product, acetyl-CoA. That acetyl group is now bound to CoA, and that leaves our lipoate molecule in its reduced form. And the acetyl-CoA goes on. This, remember, all takes place within the mitochondrial matrix, and within the matrix, the acetyl-CoA is going to be involved in the citric acid cycle. But even though we've got our 2 products, we've got our CO2, we've got our acetyl-CoA, we're not done. Remember, an enzyme has to end the way it began. Well, it started with an oxidized lipoate, and here we've got a reduced lipoate, so we have to oxidize that. But before we do that, let's just take a more careful look at what a coenzyme A cofactor looks like. What we have here is we've got this big molecule here, and then attached to it this pantothenic acid, and then we have this portion here, mercaptanoethylamine, and it's right here, this sulfhydryl group. So all of this is structural. None of this is really functional. It's just kind of holding on to the important sulfhydryl group, and it's this sulfhydryl group that is going to bind to our acetyl group. And there's another thioester right there, sulfur carbon bond. Very reactive bond. That's what's going to be necessary for the ability of acetyl-CoA to be involved in the first reaction of the citric acid cycle. All right, so now we are looking at oxidizing, reoxidizing lipoate back into its oxidized form, and that is the sole job of E3. It has a catalytic cofactor called FAD, which oxidizes lipoate, and in so doing, it becomes reduced. But now we got a problem. We started with a oxidized FAD, and now we've ended with a reduced FADH2. We need to reoxidize it to reset this enzyme properly. That's the job of our other cofactor. In this case, it is a stoichiometric cofactor or a co-substrate cofactor, NAD+. It starts off oxidized. It oxidizes FADH2 back into FAD, and in so doing becomes NADH. Now we've seen NAD plus NADH before, but we haven't seen FAD. Let's take a closer look at FAD. FAD is flavin adenine dinucleotide. Okay, so it's very similar in structure and function, in fact, to NAD. The difference is that NAD accepts 2 electrons and only 1 proton. FAD accepts 2 electrons but 2 protons. So let's take a look at that. Here is FAD, and this is something that you are going to, so this is the nucleotide portion of FAD, and then here's the part that is going to accept those electrons and the hydrogens. So first, right here at this end, it accepts 1 electron and 1 proton, so essentially an entire hydrogen atom. Boom. And then right over here, we accept another proton and another electron. Again, essentially an entire hydrogen atom right here. And so what this leaves us with is FADH2, no charge. Whereas NAD plus, right, carries a plus charge when it is oxidized, and then NADH, again, carries no charge when it is reduced. Okay, so that allows us to have a reset enzyme. After the action of NAD plus oxidizing FADH2 back into FAD, we've reset the enzyme. We're ready to go again. We've got our 2 products, CO2 and acetyl-CoA. Now, why is it that we have this PDH complex, this E1, E2, E3, multiple copies of each in this giant complex? Well, what that does is it keeps the cofactors together. Right? This swinging arm of lipoate is dependent on being at the right place with TPP, exchanging that acetyl group. And this swinging arm again of lipoate, which is swinging now over here to E3 to get reoxidized, again, depends upon them being close together. And so being in a complex ensures that all of these cofactors will be close together. It keeps transient and highly reactive intermediates from being lost and reacting with incorrect other compounds. Okay, so these thioester bonds are pretty reactive, and we want to make sure they're only reacting with the things that they're supposed to be reacting with. Being in a big complex maintains a local high concentration of reactants. And finally, this giant complex can be controlled as one. It's much easier to regulate it. And speaking of regulation, let's take a look at some of the factors that go into regulating the PDH complex. Now, the type of regulation that we're talking about is allosteric regulation, meaning that each of these molecules that are essentially a product of the PDH complex are allosteric, in this case, inhibitors of the enzymes in the PDH complex. So basically, things that would inhibit the PDH complex are things that are indicators of high energy, which means if you've got a lot of NADH, if you've got a lot of ATP, if you've got a lot of acetyl-CoA, you've got plenty of energy, and the cell does not need to make any more high energy compounds or break down any more high energy compounds. And so these molecules then are allosteric inhibitors of the PDH complex. Now, a low energy charge, so if you've got, instead of a lot of ATP, you have a lot of ADP, and if you have a lot of pyruvate sitting around that's like, hey, we've got plenty of our reactants, we can go ahead and break them down, then those are going to be allosteric activators of the pyruvate dehydrogenase complex. They're going to bind to that PDH complex and activate it so that we break down more pyruvate, that we build up more ATP. Okay, so again, go back, watch this video, watch the other videos that are involved in that describe pyruvate decarboxylation. Make sure you're able to do this, that you're able to answer these questions. What are the components of the pyruvate dehydrogenase complex, the 3 enzymes? What are their names? Where in the cell is this PDH complex located? What are the products and the reactants of pyruvate decarboxylation? And then for each of the 5 cofactors in the PDH complex, basically make sure you know the full name, which enzyme they're part of, recognize their functional part, and recognize the structure, what that functional part carries, and then whether it's a stoichiometric or a catalytic cofactor. List the reasons why there are enzyme complexes and how the PDH complex is regulated.